Hello and welcome to another episode of Teach Me in 10. Today we're joined by Dr. Ben Greulich, an assistant professor of biology at Mercer University. We'll be exploring the field of epigenetics, what it is, how it happens, and why it matters. Stay tuned to understand not just what epigenetics is, but also how it shapes gene expression and impacts disease. Hi Ben, thanks for joining us today. Could you begin by explaining what epigenetics is, and can you discuss some classic examples where we may see its effects?
So epi, really the stem of the word means on top of. So epigenetics means on top of genetics. And so what this is referring to is its modes of inheritance that are on top of or in addition to genetics. That's to say, things that are outside of the actual DNA sequence, the sequence of the letters of your DNA. And so frequently, this is going to be chemical modifications to the DNA itself, but it can also be, and frequently is, modifications to proteins that are called histones that the DNA interacts with. And so epigenetics influences how genes that we inherit are expressed. An example in humans of epigenetics, and also other mammals, is the X chromosome and its inactivation. So for example, in humans, females typically have 2 X chromosomes, while males only have 1. And it turns out if in females, both of those X chromosomes get fully expressed, you get too much gene expression of those genes on the X chromosome, and it can cause a lot of problems with gene dosage. And so the way the cell gets around this is that those cells in females will epigenetically silence one of those X chromosomes and keep it permanently turned off, permanently inactivated. Another classic example of epigenetics can be seen in Angelman syndrome and Prader-Willi syndrome. So there's a collection of genes that are epigenetically silenced in the formation of male gametes, and then other genes that are epigenetically silenced in female gametes. So normally, an individual would get one set of all of the genes from each parent, so you would have at least one set of those genes that would be expressed. But sometimes you get regions of the genome that get deleted. And if that happens in this particular region, it'll actually depend, the outcome will depend on if the deletion happened in the male gamete or the female gamete. Right? So if the deletion happens in a male gamete, it produces a syndrome called Prader-Willi syndrome. But if the deletion happens in a female gamete, it produces a completely different syndrome called Angelman syndrome with different phenotypes. And it's really interesting because it shows that while normally the parent you get an allele of a gene from shouldn't matter, but there are cases like this where the sex of the parent influences how that gene gets expressed.
So you mentioned there that epigenetics involves chemical modifications to the DNA and histone proteins, but can you explain a little deeper how this actually happens through epigenetic mechanisms?
So epigenetic mechanisms can come in lots of different flavors. There's many mechanisms of regulation at the epigenetic level, and they have varying effects. So as we mentioned earlier, commonly what's happening are these proteins that DNA is associating with, these proteins called histones, become chemically modified. So 2 of the classic examples are methylation and acetylation. And so these marks can change how tightly compacted DNA is. And so if the DNA becomes really compact, if it really squishes together a lot, it can hide or obscure DNA, and that makes it inaccessible for enzymes that are responsible for reading DNA and expressing it to be able to access it, right? It can't reach the DNA, and so if it can't reach it and it can't read it, it can't express those genes. There can also be marks like these histone marks that serve as these molecular landing zones for proteins that can either promote or inhibit expression of genes. And so you can regulate gene expression epigenetically that way as well.
So you've explained that chemical changes to DNA and to histone proteins across the genome can alter how accessible genes are. We've all heard of the genome, but what's an epigenome?
The epigenome is kind of the entire collection of all of these epigenetic marks, all of these chemical modifications that go in regulating the expression of our genome. And it can be influenced by a lot of things. It can be influenced by the environment in a lot of ways. One way is diet, right? So a healthy diet, we should be getting our vitamins and minerals in that diet. But really, what are vitamins and minerals? Why do we need them? We need them because they serve as cofactors and coenzymes, which means that they help enzymes do their job. Without them, the enzymes fail. And so that's true for these enzymes that lay down or remove these epigenetic marks. So if you don't have the proper vitamins and minerals that help these enzymes lay down the methylation marks or the acetylation marks or remove them, you're going to have a problem with your epigenome. Stress can also play a role, whether that's internal stress or environmental stress like pollution. Stress and chemical toxicities, they can change gene expression of lots of different genes, including genes that are important in epigenetics. So for example, BPA, a component of plastics, that has been linked to reducing the expression of genes that add methyl groups to DNA, these DNA methyltransferases. And if you reduce the expression of those enzymes, you're going to get less DNA methylation, which leads to a phenomenon we call hypomethylation. And that affects the expression of those genes that are being hypomethylated.
So you mentioned there that these chemical marks don't act in isolation, but occur across many regions of the genome and can be influenced by factors like diet and stress. What are some of the applications of studying epigenetics, and what challenges does the field face?
Epigenetics is a big field of study in terms of human health, particularly in cancer biology. So cancer tends to have global hypomethylation, where if you look across the whole genome, you have less methylation marks than you normally would have. But there are these regions throughout the genome that are actually hypermethylated. You get way too much methylation. And those tend to be in the promoters of tumor suppressor genes. So these are genes that help prevent tumors and cancers from forming. And when you hypermethylate, what that does is it typically silences that gene. So the cancer is silencing these tumor suppressor genes that allows the cancer to go on and develop. Cancers can also have altered patterns of histone methylation and acetylation, which again leads to dysregulation of gene expression. Because of the importance of epigenetics in cancer, it's become a really big, important field for scientists to study how can we manipulate this to treat cancers where we have these problems. And it's a really exciting field. There's a lot of potential there. There are already epigenetic drugs out there, like there are HDAC inhibitors. So HDACs are histone deacetylases. So they remove acetyl groups from histone proteins. And those have been shown to be beneficial in certain cancer types already. The biggest challenge for this field, though, is specificity. Because as I mentioned, cancer often increases an epigenetic mark at some locations, right? It can increase methylation marks at those tumor suppressor promoters, but it might decrease that mark elsewhere in the genome, right? It's not like one consistent mark applied across the entire genome. But these drugs, these epigenetic drugs, typically create a global increase or a global decrease in that particular mark, right? So an HDAC inhibitor inhibits histone deacetylases, right? And so that's going to be true across the genome or across your epigenome. And so that makes them not super specific. We're kind of hitting all of these genes when we only need to hit a few of the genes. So figuring out a way to specifically regulate those marks at certain genes, but leave other genes alone, would be a really huge victory for the field of epigenetic research.
Many thanks to Ben for joining us for this Teach Me in 10. Hope you enjoyed. Bye for now.